Welcome to the ideaXme show, the show that profiles important humans behind the really big ideas that shape our world and inspire future creators, and for all those that love really big ideas and great stories. And I'm Ira Pastor, your longevity and aging ambassador. And today we're going to continue our journey through what I refer to as the biological architecture of aging, and take a trip down to the level of the genome. But we're not going to talk about the traditional, simplified, static, reductionist genome that the majority of the public is usually treated to. Today we're going to talk about a complex genome, one that takes into account that the genome is a very fluid and highly networked place filled with endless feedback loops, hierarchies of control, intricate phenomena like pleiotropy and epistasis and alternative splicing, and a genome that is continually interacting with its microenvironment, other genomes, metabolic architecture of cells, other individual species, yielding wide varieties of traits. Today, I'm really pleased that we have Dr. Michael Rose joining us. He is, without a doubt, one of the leading scientists in the area of aging, biologic immortality, human evolution. He's a professor in the Department of Ecology and Evolutionary Biology at University of California, Irvine, and for the last 40 years has been active in studying biologic aging via techniques of experimental evolution. And he is truly the preeminent thinker in the areas of complex theory, such as declining forces of natural selection, antagonistic pleiotropy, mortality rate plateaus, author of hundreds of papers and numerous books, including the Evolutionary Biology of Aging, Does Aging Stop?, Methuselah Flies, The Long Tomorrow, Darwin Spectre, and ultimately a really wonderful synthesis that I encourage everybody to go and read called The 55 Theses, 55theses.org, where he truly distills down a lifetime of knowledge in 100 or so pages into a very readable format. So with that introduction, Michael, thank you so much for joining me today on the show.
Well, that was such an awesome introduction. I can't imagine the interview really improving on it.
Thank you. Thank you. So we usually start off this show by just giving the guests the floor. For those that are unaware of you, would love to hear about you, your background, where you grew up, how you got interested in science, how you got interested in evolutionary biology, and ultimately how you got interested in the topic of aging.
Okay, so I'm a military brat born in a military hospital in Germany in the 1950s. My father was an early NATO officer and Canadian NATO officer, and I spent my childhood being dragged around Europe, Canada, and the United States because of my father's military career. And then I went off to college at 16 in 1971. And then I spent about 16 years going around the world between Europe, Canada, and the United States, developing my scientific career until I ended up at the University of California, Irvine, 32 years ago. And have stayed ever since for a host of reasons. I became interested in science. The interest started when I was 7. I can remember when I was 7, one Christmas, that Christmas when I was 7, being given a microscope set, like a real microscope set, like a compound microscope by my great aunt with glass slides and forceps and tweezers and scalpel, and I opened it up, and I was completely excited, and my parents saw what I had, like a scalpel with blades, and they immediately took it away from me. I didn't get it for another year and a half. So, but and perhaps tragically, perhaps fortunately, because I couldn't indulge my immediate interest in creating slides and looking at them through a microscope, I satisfied my growing interest in science by astronomy and physics, which became my primary scientific obsession for between the ages of 8 and 10. And, so when I was 8 or 9, I got a telescope. My parents figured that was less dangerous than a microscope. And when I was 10, I built a planetarium. I was from a kit. I don't think I built it from scratch. And when I was 11, I remember reading books on theories of cosmology. Though I'd been interested in theories of cosmology since I was 9. But starting when I went to high school, I was 12. Cue the young Sheldon jokes. I started to drift more and more toward biology. And by the time it came time to go to college, I was really a biology major. And then I was already interested in evolution. Before I went to college, I had a fantastic course on evolution when I was 13. But I really fell in love with evolutionary biology as an undergraduate. I started doing research in 1973. And that's now 46 years ago. And an important part of my background that most people don't know about is much of my early scientific training came from engineers doing systems analysis of large-scale simulation models. So we had a dedicated computer, like that filled up our
Sure.
did large-scale simulation projects on. And I became interested in relatively esoteric things like how you put data into a large-scale computer simulation model, how you do sensitivity analysis, how you derive more simplified models. I became interested in nonlinear dynamical systems mathematically, so that I wanted to do theory as a graduate student. And my first graduate degree was indeed biological theory, primarily. Then I wanted to do a PhD, and one lab at Harvard, then famous evolutionary biologist called Richard Lewontin, was perfectly happy to continue indulging me in my interest in theory. The other lab I wanted to go to was that of John Maynard Smith and Brian Charlesworth at the University of Sussex. But John Maynard Smith, who did some really foundational work on aging in the 1950s, said, no, Michael, you can't do your PhD on theory. You have to do some experiments. And the experiments that he and Charlesworth wanted me to do were on aging. And between 1975 and 1976, they talked me into doing a PhD on aging when I thought it was a terrible idea. Because in the 1970s, through the whole decade, aging research still had a tremendously bad reputation. Credible scientists didn't do it. Most of the people who did it were MDs who were kind of amateurish scientists rather than, well-trained research scientists. John Maynard Smith had been an exception in the 1950s and 60s. But much of his work revolved around destroying one cellular or molecular theory of aging after another. He was a complete wrecking ball. And I frankly, I admired him for that because he was a Popperian, follower of Karl Popper. Falsification is the most powerful tool in the experimentalists' armamentarium. And I also was a Popperian before I ever met him. And I thought that was beautiful. Although the irony of the situation was that Brian Charlesworth, my other advisor, wanted me to prove by some experiments that his favorite model of aging was correct. And his favorite model was something that was called mutation accumulation, which is a misleading term because it was not somatic mutation, which is the way that term suggests aging is caused. No, it's an evolutionary process of accumulating bad mutations late in life based on the mathematically well-founded view that bad things that happen at later ages won't matter,
Sure.
which I think is fundamentally correct. But there was a subtle aspect to that theory, which was that it only worked if the genes affecting the different ages were basically only age specific. Unfortunately for that theory, if there's anything that we have learned about the physiology of aging and the whole life cycle itself, is it's interconnected by pleiotropies, positive ones and negative ones. And I invented the term antagonistic pleiotropy for the negative ones, where you have a good effect, bad effect on another good effect. So off I go in the lab in 1976. According to Brian Charlesworth, I'm supposed to be showing that his theory is right. They do experiment after experiment, and the results always show that his theory is wrong. And I further should emphasize that he convinced me that he was right before I started doing these experiments. Only all the results came back showing that he was wrong. Now, I was enough of a Popperian to think to myself, before I started doing these experiments, I should design them so that I can not only tell whether or not his theory is right, but also whether or not his theory is wrong. And furthermore, whether or not the other major genetic idea about aging, which is antagonistic pleiotropy, is correct instead, or wrong. Maybe all those theories were wrong. Maybe the whole thing, because they're all tests of the evolutionary theory of aging. Maybe all my experiments show that everything was wrong, just as John Maynard Smith had spent 15 years of his life showing that all the cell molecular theories that had been proposed at that time were wrong. And instead of showing that they were all right or all wrong, the evidence really came down strongly in favor of an antagonistic pleiotropy explanation of aging. So now let me try to explain, for those of your viewers who have not weathered this terminology before,
Sure.
Go ahead. What it means is an idea that not original to Brian Charlesworth or myself, an idea that had been kicking around since the 1940s. Whether or not evolution sacrificed later survival and reproduction for the sake entirely of earlier reproduction. Whether or not that was at the foundation of aging. And I have spent all these years since 1976 accumulating evidence that that theory is almost always the main evolutionary explanation. Under very exceptional conditions, you can contrive things, and we've done this in our lab. You can contrive things so that mutation accumulation can work. So mutation accumulation can work in some small fraction of cases. Overwhelmingly, it's all about antagonistic pleiotropy, which is to say evolution can build immortal organisms, and in fact, it has already done so. Any number of single-celled organisms are immortal,
sure,
as long as they reproduce by splitting into, and there are lots of plants, from weeds to trees, that have no aging, and there are some animals that have no aging. And my favorite example of that is sea anemones, but it's also been shown in great detail in Hydra. They're both coelenterate, but they're multicellular differentiated animals, all the same.
Right.
The same cells we do, telomeres, mitochondria, nucleus, all the same equipment. Yes, they have free radicals. Yes, they experience free radical damage, all those things. But evolution basically says, no, we don't want them to age, so they're not going to age.
Right. And
the details in that are fun to talk about, because immortality is my second biggest interest after aging.
Sure.
So now let me explain a little bit about the sort of classic experiments that I've done that are I think, pretty simple embodiments of the ideas I'm talking about. So if aging is something that evolution can establish or banish whenever aging cares, the question is, well, what makes aging care?
Mm-hmm.
And what makes aging a matter of indifference to evolution? And the key concept was first proposed in 1941 by J.B.S. Haldane, one of the great evolutionary biologists. And he basically proposed verbally that the force of natural selection declines with adult age. In 1966, another truly great evolutionary biologist, William Hamilton, did a mathematical analysis of the force of natural selection and showed that in organisms that only reproduce by having sex, like humans and insects and all mammals,
Mm-hmm.
that inevitably after the start of the reproductive period, not the end of the reproductive period, at the start of the reproductive period, the force of natural selection acting on survival starts to decline,
Mm-hmm.
and then it declines all the way to zero and stays at zero forever after. And Brian Charlesworth, one of my doctoral advisors, further showed that if you add genetics to this picture, Hamilton is fundamentally right. If you add genetics to this picture, and Charlesworth did a lot of very complicated heavy lifting to show that in more complicated genetic systems, Hamilton's originally crude model still applied. Brian Charlesworth did some very pretty mathematics that showed that if mutation accumulation was the genetic foundation for the evolution of aging, then certain elegant consequences would follow, or things like the additive genetic variance of fecundity at specific ages. And I'm not going to go into that in detail because it turns out his model is almost always wrong. But in my research, what I showed is the following. If you increase the force of natural selection at later ages, natural selection will then, in a lab, like
Mm-hmm.
make evolution slow down at earlier ages, and we can even drive it down to the point of stopping it at early adult ages so that it only starts again later. It starts up really for the first time later.
Sure.
And the giveaway that this was due to antagonistic pleiotropy is that when we slam down early and middle aging, what also happened was that early reproduction got reduced. And what does evolution by natural selection love more than anything else? Early reproduction. We should say that evolution loves trailer park trash.
Mm-hmm.
So, you know, Cleon from Idiocracy, natural selection smiles on Cleon, and Carol and her late husband from Idiocracy are not the kinds of people that evolution by natural selection likes,
right,
normally. So, of course, my game was to reverse those rules and say, you know, to the flies, fruit flies in my lab. I don't have a lab that's, you know, people are the subjects.
Sure.
If you go ahead and reproduce yourself to death at early ages so that you're washed up by middle age and soon going to die, you don't get to contribute to the next generation because I'm going to discard all the eggs that you laid if you were a female or that you fertilized if you were a male. And I'm only going to keep the eggs that were produced at later ages.
Mm-hmm.
And all I'm doing with that procedure, right, is I'm slapping evolution by natural selection upside the face and I'm saying, okay, you got to do something about this early to middle life aging period, right?
Right.
You want to clean that up. Can you do it? And the answer is, it certainly can. It can do that. So I basically had that result more than 40 years ago.
And that's a wonderful segue now into 1981, where you begin conducting, you know, what is this longest running experiment in Drosophila melanogaster experimental evolutionary breeding, producing these organisms with 4 times the original lifespan. Let's get a little into that work, some of your findings, and specifically, because we'll separate this out, because we'll talk about genomics in a little bit, but if you could sort of go into a little bit of sort of the physiological findings in terms of fitness and fertility, robustness, resilience that you find in these organisms that have been mastered.
So let me give credit where credit is due, and to say that virtually every insight I've ever had about aging, beyond the basic force of natural selection idea, which isn't mine, it comes from Haldane and Hamilton. Every insight I've had has been handed to me by these flies. So, I don't have spectacular a priori insights like Aubrey de Grey, from the mouth of God directly to his ear. Mm-hmm. I'm not that brilliant. Instead, what I do is I try to set up experiments where I give the flies the chance to tell me how they did it,
right.
How they became so awesome. How is it possible for them to live so much longer? And they don't, and so an immediate question is, do they live so longer by totally shutting down reproduction over almost their whole lifespan? And the answer is no. What they do is they diminish the very early intense peak of reproductive activity. So you may remember what you were like in your last years of high school and college, your later 20s, right?
Right.
You were obsessed.
Absolutely.
Yes, and you're obsessed with a particular thing. And in being that way, regardless of your gender of preference, you were like the vast majority of younger males, right?
That's it.
Yeah. So I remember those days. They were a very long time ago for me. And then you sometime after 30, you recover your sanity. Okay? So in these flies, that totally insane period is removed. So in these flies, instead of like going crazy at football games or after football games like Cleon and his son, Cleon Jr., Mediocrity, after the age of 30, you set about meeting an important person or persons, and you start biological reproduction, and then you take care of your kids, and you change diapers, and you take them to school.
Oh, yeah.
So the flies come back and tell me, no, we're going to reproduce more in total than, the normal flies that go crazy really young, really fast, and then age off to death and oblivion and infertility fast. We're going to live totally differently. We're going to show reasonable levels of reproduction, and we're just going to sustain reproductive activity for a very long period of time. And, so full disclosure, I had my last child just before my 60th birthday.
God bless you.
So, and I was also totally celibate during high school. So, before I even knew the results, I lived by the results. So it's like I was the opposite of Cleon. I was, total nerd boy carrying around my big briefcase full of books with my thick glasses. And my 2 best sports were chess and quiz show. So I was a nerd's nerd. So basically, these flies are nerds.
Gotcha.
They're not like the cool kids in high school doing drugs, competing in team sports, and having lots of sex. They're like the way I was in high school. Completely boring, totally unexciting, and so on. Mm-hmm. That's the first and somewhat surprising lesson. Really, that they're stopping with the really hot teenage equivalent, early adulthood years. They're being like well-behaved, nerdy kids who will go on to get PhDs and MD. People who listen to this interview are exactly like what I'm describing.
Probably.
Not all, but probably the clear majority. Yeah. So the second thing that I learned was a complete case of serendipity. Without going all the details. One day, my then only lab tech decided to do an amazing experiment. Well, she didn't really decide. She forgot to feed the flies for a period of about 24 hours. And I show up to the lab the next morning, and she's at her desk having a complete nervous breakdown. Her face is buried in her hands. She's crying, the whole bit. I sort of go, what's wrong? She doesn't even look up. She just points at the incubators that have the flies in. So I go over to the incubators. I open the first door, and I look in at 2 cages of flies. I immediately notice there's no food in the cage. It's like, oh, I think she forgot to feed the flies yesterday. Then I look more carefully. In one of the cages, the flies are almost all dead. And in the other cage, the flies are almost all alive. And these 2 cages contain flies of the same age. Okay? And then I notice how they're labeled. The cage with the almost all dead flies is our control population, the ancestors of the flies that evolved longer life, but they didn't evolve longer life. They're the controls, the normal flies. The cage where the flies almost completely survive a prolonged period of no food are the longer lived flies. So I start smiling, and I pick up these cages, I walk them out, and I put them down on the lab bench, and I just stare at them. And all of these experiments are rushing through my mind, all these experimental designs, experiments I want to do. And my technician looks at me, and she figures like I've gone completely crazy. Right? Because she doesn't understand she's done an incredibly brilliant experiment that I wouldn't have thought of doing for years. And it opened up a whole new line of research, which is the relationship between acute stress resistance and aging. Mm-hmm. Mm-hmm. And it turns out that when evolution builds a much longer lived organism, it doesn't make a feeble organism, it makes a more robust organism.
Excellent.
And it's more robust even at early ages. And then middle age, and of course at later age. And it's just like completely better at resisting total starvation, total desiccation, high levels of ethanol. So it's like perfect for living in Las Vegas, where you go from casino to casino when you're dry out and you're drinking too much. And so it's like a Las Vegas fly. So we then spent years working on the relationship between stress resistance and aging. Mm-hmm. It's a very close relationship. And we went deeper into the physiology, and we basically discovered that if you want to live longer and resist acute starvation, you will be moderately fatter, but not really fat. You basically won't be scrawny. If you want to resist desiccation better, you're going to have better water reserves if you're fruit fat, and lose water at a slower rate. So it turned out that, and this surprised me, this was work primarily done in the 1990s. It surprised me because aging was very clearly bound up with essential generalized aspects of physiology. It wasn't a very specific thing. Mm-hmm. It certainly wasn't molecular in the sense in which the term has been used primarily over the last 20 or 30 years. It's instead sort of basic aspects of how you build an organism. Mm-hmm. You want to make a much longer lived organism, you can change some fairly simple things, which in turn, frankly, I think is very exciting from the standpoint of intervening physiologically and not evolutionarily or genetically in the physiology of any organism you would like to live a longer and healthier life, like they may be humans. Mm-hmm. Okay? Like maybe you care more about humans than fruit flies. It's possible.
It's possible.
It's possible. So if changing aging doesn't require that you do a phenomenally expensive RNAi intervention, mm-hmm. costing, hundreds of thousands of dollars for every individual whose aging you change. Instead, if the manipulation of aging by non-genetic means requires that you retune sort of basic or housekeeping aspects of physiology, then the prospects of finding ways to intervene in human aging become far better.
This is fascinating, and when I hear about these findings in terms of fertility and fitness and robustness, as you're explaining it, that we now drill down to an era we'll call the omics era. We call it genomics, proteomics, or transcriptomics, whatever you want to put in there. And listening to some of your talks in the past, I'm always fascinated by sort of this, I don't know if you call it an epiphany that you had, but because you're so into the data and the information that flows from this physiology. And then, you're always there explaining, hey guys, I'm looking at this, range of information at all these levels. And you know what? It's not about telomeres. It's not about this tumor suppressor gene or this DNA response or this heat shock protein. It is literally waves of activity across hundreds of sites of the genome that are fluctuating and sort of playing together in this symphony. And so I want to sort of delve a little bit more into that theme, because I think there are some very, this sort of the systems biology folks refer to that stuff as, attractor states of the genome, and how little changes can really ripple through the genome and move physiological state in one direction or another. But if you could sort of dive more into your thoughts and feelings about those learnings, I think that would be really interesting for the audience.
So again, I have to credit the flies, right? So everything I'm going to say about this topic, which has always been of interest to me, before we had genomics, I was interested in genomics, as it were. So in around 1980, when I was designing my system from the ground up, I wanted it to be able to look at many sites in the genome. So for example, I published a paper in 1993 with a man called Jim Fleming, which used 2D gel electrophoresis of proteins, which is a horrible technology. It's very hard to get it to work. You don't know what the proteins are. Everything is tough. But we used it to try to get a sense of, how much of the genome was changing in order to produce a longer lived fly. And the answer we got then was at least 2 to 3% of the functional genes were changing. And that was a minimal estimate for several reasons. One is there's not even that much protein production in an adult fly. It's very, very limited protein synthesis. But anyway, and we used adults. So I took that 2 to 3% figure, and I ran with it. And by the year 2000, I was trying to do some type of omics. And the 2D gel technology was like proteomics, caveman proteomics, by the standards of what we got. So with the advent of the genomic era, of course, there were 3 organisms where we learned a lot about their genomes really early. And those were firstly C. elegans, the worm, and secondly, Drosophila melanogaster,
sure,
and third, the human. And as I describe in The Long Tomorrow, I went, aha, well, that means that if we can find a genomic matchup in between longer-lived fruit flies and humans that resist chronic aging-associated diseases, we have bridges between the flies and the humans, and we can use the places where we have bridges to focus our analyses for transforming human aging, whether pharmaceutically or by some other type of intervention. So my bet going into that work was that a few percent of the fly genome would turn out to be important, and that maybe 50% of those genes would also be in humans, and that maybe some still smaller of that stuff would turn out to be associated with human aging-associated diseases using techniques like GWAS. And therefore, I could go from, fly omics through human omics down to, 10 or 20 candidate sites in the human genome that also map back into the fly genome for like drug development. And I expected like maybe 2 or 3 of those genes would be associated with fat metabolism, 3 or 4 might be associated with, genetic integrity or something like that, okay? So I was thinking in terms of fundamentally there being hundreds of loci involved, and that what we could tractably identify and work with would be on the order of dozens, okay? And I worked away in that project at the university and companies, back and forth. And it turned out that I was wrong, but I was the least wrong, because everybody else I talked to was thinking in terms of are eventually finding somewhere between 1 and like 5 or 6 genes, okay, that would matter. No. The correct number of sites in the fly genome that are probably involved in aging is at least 1,000 regular genetic loci together with other sites that don't make regular genes but are all nonetheless transcribed with functional effects.
Sure.
You got 1,000 genomic sites. You have at least hundreds of transcriptomic differences, and we've just started working on metabolomics, and the number there is probably going to be high too. Then, okay, so you might go, yeah, but how useful are flies to human results? I was one of the more skeptical people on that question. Well, it turns out I was totally wrong about that too. Fly data and human data say, no, you got like an 85 to 90% overlap.
Yep.
Specifically for the genes that we implicate as being involved in aging. Flies, it turns out at the genomic and transcriptomic level, are amazing models for the physiology of human aging and chronic disease. So now instead of the problem being having too little at the end of this process of going back and forth between fly and human genomes, the problem is instead too much.
Right.
The problem is instead, thousands of genomic sites, thousands of transcripts, who knows how many metabolites to worry about. Both systems.
Right.
Right? That's what GWAS of human chronic diseases overwhelmingly shows. Lots of sites to worry about. It's hard to identify them in humans, harder than it is, of course, in live flies, which is why the basic idea of going from flies to humans is still a very powerful one. But it's like 2 orders of magnitude more complicated than I thought, 3 orders of magnitude more complicated than everybody else thought. I was still wrong. The flies told me, you're wrong, but you're less wrong.
Hey.
And less wrong is sort of my new motto. Is I'm all about being less wrong. If I can be the least wrong, I'm very happy.
Nothing wrong with that.
So there we go. So that's why over the last 7 years, our biggest project has been the integration of the whole biological hierarchy from genome, transcriptome, metabolome, physiology, life history, aging, the whole package in terms of the functional side. And then using AI to penetrate this enormously complex hierarchy. So we published our first AI analytics paper a little under a year ago in the spring of 2018. And we have tons of stuff coming down the pipeline with much larger bodies of data. Turns out that you need to have approaching 100 populations of data to attack this problem. And you have to have populations like we have in our lab,
right,
where you have many highly differentiated populations. So back in 1980, I thought sort of 5 longer-lived populations and 5 controls. Well, that would be cool and powerful. But that was from a GLM, generalized linear model, 20th century data analysis standpoint. Once you're in the world of omics and AI, that just does not cut it.
Right.
You have to have the kind of population and omic data that right now, to the best of my knowledge, only we have. No other lab in the world has what we have.
Excellent.
Public sector or private sector doesn't matter.
I'm glad someone has it.
And the only way you can parse it is with AI.
Got it.
You can't tell a story. You know,
Sure.
you can't tell a story about 121 sites in the genome that modulate heart function.
Absolutely.
And frankly, you can't even use GLM. You can't say, you know, the correlation or the regression of heart function on the genome is like this particular number. It's
Right.
not like that. You have to use AI tools.
Right. Well, stepping into AI and futuristic technologies, thinking of next iterations and so forth, but jumping over for a minute to sort of where you are, and then the 55 theses. So, you know, I've read through them. As I said, it's a wonderful read. I encourage everybody to download them and really get some insight. You make some really important scientific cases along the way for various actionable things. You know, people question about dietary and lifestyle interventions and so forth. But I want to talk at a slightly different direction. You know, coming off of the sort of the next steps. And you know, in your thesis 55, you basically you state as follows. You say further changes moving forward in the future would enhance human health at later ages. Will be discovered using evolutionary research tools, experimental evolution, model organisms, and the further molecular genetic analysis of human evolutionary history. Then you talk about a revolution in human health. I love your statement. The cold, gray grip of the reductionist biomedical establishment will be progressively weakened by the onslaught of reductionism killing genomic data. I love that. And as I said, I want to go in a slightly different direction than you normally take in on this and have a little fun, because moving forward, I want to imagine a scenario for a moment where you get a phone call. Dr. Rose, it's the CEO of Pfizer, Merck, J&J, whichever big pharma. And they admit to you, you know what? We've been doing this wrong the last 100 years. We know this.
This is science fiction. This is fantasy.
Fantasy, but let's have some fun with it. We are going to we're going to give you the keys to the kingdom. We're going to give our $5 or $6 or $7 billion research budget for the next drug development cycle, 15 years or so, to you. And you are going to cure aging and come up with these health span, lifespan related interventions. Now, I understand you're not a drug development translational guy in that context, but I want you to just talk about a future scenario that you see. You can do anything you want. You can develop drugs, nutraceuticals, functional foods. You can work with small molecules, proteins, gene therapies, electroceuticals, invent stuff that hasn't been invented yet, per Acacia. Anything you want to do. What does the next 15 years look like using some of these tools, bringing the AI in, bringing the newer types of therapeutic interventions, that brings us to the year 2035, 2040, and so forth, that everyone's so excited about, that is the time where we're supposed to reach some type of escape velocity or biologic immortality. Take a run with that one.
Okay, so I've basically been thinking about that challenge for about 30 years, because about 30 years ago, I was contacted by a VC, which was in principle suggesting I do something like that with their money, or however much money they could raise. And so I've been developing plans or answers to that query for 30 years.
Okay.
And it turns out it's a large and complicated project that involves different things. I separate out those different things into 4 main bins in an article called 4 Steps to the Control of Aging, following the example of infectious disease. Because basically, the world asked Louis Pasteur that question, 70s and 1880s. And he had lots of fantastic answers, which the medical establishment of his day resisted every step of the way. Right? was in 1840.
Okay.
The medical establishment didn't really fully accept it until sometime between 1910 and 1920. So for 70 to 80 years, hundreds of millions of people died because they resisted the germ theory of disease. The evolutionary theory of aging was basically proposed in 1941. And now, about 80 years later, the biomedical reductionist establishment still resists it. The number one most important thing that has to happen is the reductionist biomedical establishment has to give up on resisting the truth about 90% of what goes on in health.
Sure.
There is, on the order of 5 to 10% that's all about specific genetic diseases for which precision medicine, as they now call it, would be perfect. Huntington's chorea, sickle cell anemia, cystic fibrosis, Tay-Sachs disease. Go to town, guys.
Right.
This is what Francis Collins was brought up to handle. And then there's, of course, well-defined pathogens, HIV, malaria, and so on. Go ahead, hit them with all your stuff. The other 90% of deaths in advanced industrial countries, which is fast becoming the majority of the world population, right? We're dying of heart disease, complications of type 2 diabetes, cancer, dementias, kidney failure, chronic kidney disease, and heart failure, and so on. All right. So I would say to Merck or Pfizer in your fantasy sequence, great. So you can have a division which is going to continue to go after these very specific disorders with very well-defined etiologies, which really come down to one gene or one pathogen. And it should be said, and tragically so, that those things are characteristically not very profitable because you spend vast amounts of money to cure small numbers of people, and often those cures themselves are very expensive and arduous.
Right.
So what I'm all about is the 90% of all the major causes of death and disability that you and I face, that you've seen in your parents, grandparents, and so on. It's tragic and heartbreaking. And what I'd say is, now that I've got your attention, now that you have understood that this is 90% of the whole game, and more than 90% of all the potential profits they could make, now let's go down a very different road. And just as with Pasteur, the most important changes to be made immediately have nothing to do with pharma or biotech. So what's the two most important changes that Pasteur established were physicians washing their damn hands, which Semmelweis had fought for, and surgeons coming up with clean operating theaters, which Joseph Lister started to implement because of the advice of Louis Pasteur. Those took effect immediately. Somewhat secondary to that was clean water, water that wouldn't have cholera in it, for example, but any number of other pathogens in the water. And then pasteurization of milk. Hello, where did we get that name from? The man who saved more lives than any other human being on earth, Louis Pasteur.
Absolutely.
So the second step is totally ordinary things you can do with your life. And the best thing that evolutionary theory shows, and the flies now confirm that this is correct, is the older you get in biological time, the farther back you have to go in evolutionary time. The most extreme example of this is foods that were invented in the 50s, 60s, and 70s, and lifestyle components that were adopted only in the 20th century. So spending almost your entire day sitting or lying down, and I include under the sitting category behind the wheel of an automobile, traffic in Los Angeles, and eating Twinkies, Red Bull, Coca-Cola. Nobody's adapted to that. That is an evolutionary disaster. That's what evolutionary biologists like to call a mismatch. those are total environmental mismatches. Nobody at any age should be consuming high fructose corn syrup. Nobody at any age should be consuming seed oils, which only became part of the agribusiness system in the late 1800s. And then only in the United States. They only got to Europe after 1900. So that's the second step. And in the case of so that actually this second step may sound like very hokey. It sounds like just like dumb paleo ideas. There's actually some deep mathematics underlying this, which someday will be able to get published despite the resistance of even evolutionary theorists to the mathematics. And we also have experimental data that show that if you tune the diet of fruit fly according to its evolutionary history, you could greatly improve its pattern of aging if you, at later ages, go back to their evolutionary past.
Mm-hmm.
So you can improve the health of your children by denying them Twinkies and soda, and much less diet soda, pure poison.
Mm-hmm.
You know, they're, as long as you have control of what they do. The more subtle point, though, is that your children are very well adapted to organic agricultural diets if they have Eurasian ancestry.
Okay.
Still fine-tuning. So southeastern Eurasians are not adapted to grains,
right,
only to rice. And extreme northwestern Europeans, like the Irish and the Scottish, aren't that well adapted to grains, which is why it's the Scottish especially. I'm a lot of Scottish are quite disagreeable. And anyway, that's a whole other story. But if you're, for example, from Iran or Iraq, you're very well adapted to consumption of grains when you're a child, even when you're a young adult. But because of what the math does in terms of the diet-specific tuning of the action of the force of natural selection,
mm-hmm,
you lose that adaptation in your 30s. By the time people are in their 40s or 50s, it's over. You can't be a true omnivore anymore. You have to go back in evolutionary time before the agricultural revolution, or a reasonable Trader Joe's Whole Foods approximation.
right.
And the experimental data where we basically done this with our flies, we use literally a hokey Trader Joe's emulation of the ancestral fly diet.
Mm-hmm.
It's organic applesauce.
Right.
Before we brought them into the lab for the last 1,000 generations, they fed for centuries on rotting apples. So we do a literally Trader Joe's emulation of their paleo diet.
Mm-hmm.
It works. They get a completely different late life aging pattern. It's far more gentle. So because of so I had the basic idea for all of this around 2009.
Mm-hmm.
We did the math from 2009, 2010. By 2011, we were doing the experiments. And I basically have taken my own advice. And I won't go into the grisly medical details, but before I took my advice 10 years ago, I had some very dangerous chronic health conditions, which were resolved. So I actually wasn't that confident I would be alive now, given all the crap that was happening to my body 10 years ago.
You biohacked yourself. Very good.
I Evo biohacked myself.
Evo biohacked.
Yes. And so then having done it for myself, and then once we had the math, I did it for myself because I was trained as a theoretician 40 years ago. I can anticipate the way the math is going to go. So I did it just for myself without the math. Once I had the math in hand, I started to convince my fellow nerdy 50-something professors to try this out. I said, so if the math is correct, this should happen. And every one of my nerdy, nerd bros, if you will, who did exactly as I suggested, experienced a health revolution. In one case, I probably saved his life. In another case, I got spectacular results. Then we did the experiments, and the data were basically in 3 or 4 years ago, certainly by 3 years ago. And then, of course, I started to really put up the pressure on people like my wife, her family, other people I know, more and more people. And that's what led to the 55 Theses. was basically I was talking to a fairly nerdy bro, the guy, Rob Patterson, who helped me set up the 55 Theses website, his webmaster. And he adopted it and got exactly the same results everybody else in their 50s and early 60s have had, which is everything gets better. Mm-hmm. Cognition, emotion, stamina. If you have a specific dangerous health condition, as I did 10 years ago, it will at least get better, if not fully resolve. Now, let me just give you a disclaimer. I'm not saying the kind of dietary transition I advocate, 55 Theses, will cure cancer, resolve heart failure, prevent stroke, improve the outcome of stroke. I'm not saying any of those things. I'm saying if you have a chronic adverse health condition, this is not medicine, right. It's free medicine. It's lifestyle advice. So, if you have an obese child who eats lots of Twinkies, pounds of Twinkies a day, and drinks diet soda, and they have type 2 diabetes, and you take all that crap away from them and put them on an organic agricultural diet, you will improve their health. You're not a physician when you do that. You're just like people who wash their hands after they come home, especially if they picked up their kids at school or daycare, Germ palaces, right? You take the lifestyle advice of Louis Pasteur. You wash your damn hands, especially if you're a physician.
Yep.
You rub antibiotic over a cut, apply a Band-Aid. That very simple lifestyle advice. That's the second of 4 steps. Mm-hmm. The third step was something I didn't understand was necessary until we got the genomic results. So my original vision for how the genomics would work, which was about 20 years ago, 1999, was that we would find a handful of targets in the genome. Not because there were only a handful of causes of aging at the genomic level, but because that's all we could find. Well, genomics is so awesome and fruit flies are so close to humans that no, we got hundreds and hundreds of targets.
Right.
And we are not close to knowing exactly how that 100 to 1,000-fold network works omically. So again, I revert to the example of infectious disease. Did Louis Pasteur know the cell molecular biology of adaptive immunity? Hell no.
Nope.
Did the people who discovered the first antibiotics know the cell molecular biology of killing bacteria?
Nope.
Hell no. They found that those things worked and they followed them. Now, their problem was actually easier, of course, than aging-associated chronic disease in humans. But I founded a company with a former doctoral student of mine, Dr. Larry Cabral, mm-hmm. called Lyceum, which is based on the following idea. And you can go to the company website for a beautiful video that Larry did that explains it. Even if we don't really know what's going on, AI, together with omics, can give us a first pass at the drugs that would be the equivalent of the antibiotics and the vaccines that were used between 1870 and 1960, a period of almost a century when basically Louis Pasteur was right, but we didn't know the cell molecular details.
Right.
I mean, don't get me wrong. The foundations of all of this is cell molecular biology.
I'm nodding because I've never been I hung around the pharmaceutical industry for about 30 years, and I was never one for the belief that mechanism trumps mode. You can go into 80% of the pharmaceutical package inserts nowadays, and when you get to mechanism, it usually says unknown. So I'm a big believer in mode of action and stuff working and reversing into it. Sure, I'm totally with you.
I'll give you 2 words: aspirin and lithium. We still don't know how they work.
Absolutely.
But they're awesome. They're
Absolutely.
Great drugs. They're fantastic drugs. You might think that me being into evolution means I'm against pharmaceuticals, and that's absolutely not true. My point of view is the right pharmaceutical in the right setting, fantastic.
Yep.
And I get my children vaccinated, and we take antibiotics and the whole 9 yards.
Me too.
But, right now, the pharmaceuticals associated with chronic disorders, statins would be a favorite example of mine, but also most antidepressants and so on and so on. We really are not fully analyzing how they work and what they do. We now have the tools by which we could, and unquestionably, your generic drug is going to hit, hundreds of transcripts, who knows how many metabolites. Pretending it's one single cell molecular pathway is, again, science fiction to fantasy.
Agreed.
Ridiculous. So what I've proposed in founding this company called Lyceum, Lyceum, is that we take let's find out what works, even if we don't know why, using the most powerful omic tools, the most powerful physiological analyses, and the most powerful AI, and put it together. And, we very, very concrete plans for exactly how to do that. Now, finally, the fourth step, which I think mostly comes after 2030,
okay.
Is to actually figure everything out. And I now believe that's a doable project. But if Merck and Pfizer come to me with a $5 billion budget, I put it all into step 3 of that, which is that you guys now have revenues, whatever the number is. Let's say it's $80 billion a year.
Mm-hmm.
I think I can increase that tenfold over the next 15 to 20 years.
That was something Wall Street would like.
But the reason why, and I've had conversations with executives at Merck and Pfizer,
mm-hmm.
and there are 2 reasons why they never hire me to revolutionize their companies. The first one is, this is the most common, they have a background in 20th century biology before the omic era. And in that era, it was about, you clone human growth hormone and you put it in recombinant DNA, you amp it up, and you give it to the patients that have some type of dwarfism related.
Mm-hmm.
And that's the model. And for those people, that less than 10%, it's a fantastic model. But it's not going to solve the other 90%. And they can't really get their minds around the other 90%. And then if they don't have a background in 20th century biology and they're a business person, then they're very receptive to me. They go, oh, that's cool. And I'll have to go back to my company and talk to my scientists about this. And what then happens? All the head scientists were trained in the 20th century. They were trained like Francis Collins.
Right.
They're pre-genomic. They can't wrap their head around hundreds of things to monitor. You can only parse it with AI. The human brain can't do the job. They reject that. The scientists on their staff, they have a very concrete reason for rejecting it. They lose their jobs. There's an aphorism due to Sinclair Lewis, which goes as follows. If a man's livelihood depends on his failing to understand a specific point,
mm-hmm.
you may be sure that he will fail to understand it.
My time spent in those trenches, unfortunately, on the other side of the table, and that's why I sort of got out of there myself, was exactly that. It made no sense to throw away the potential of some of these approaches, as unusual as they may be. And we had some successes in the sense that starting back in the 90s, with the FDA came around to the concept that with the development of their botanical drug guidance, as an example, that a drug didn't have to be this pure synthetic chemical entity. It could be something that was a little dirtier, a little more nondescript. Into the early 2000s, you saw the advent of living therapeutics. I mean, I make a point. Not many people realize that FDA currently has approved leeches, maggots, and helminths for various medical purposes in the United States. I mean, here we're talking about therapies hundreds of years old, but they're back. Being they are complex, heterogenic therapeutics that combine medical device and drug and the saliva and all sorts of interesting things. So we're beginning to see that. And then obviously more recently with some of the sort of microbiome cocktails where we talk about mixtures of bacteria or electroceuticals where there's no drug at all, but a wavelength. So, I mean, I think stuff's happening. I have my fingers crossed that we're going to break through because, as you said, the old model is not going to work. And, when you have 50, 60, $70 billion of revenue and you're promising Wall Street 10% growth, you're not going to get it from the old ways of doing things. So we definitely have to come up with these new approaches. And I really love your holistic thinking to this in these 4 steps. So, we'll get to the point now of the show. We talk about science fiction, and fantasy, but let's get back to science fiction for a moment. It's a sort of the question that everybody gets asked on this show. I go into my closet over here and I pull out the time machine that I've been working on, and I Dr. Michael Rose, you can play around with this time machine. You can go anywhere to the past or the future you want. You can go talk to anyone you want. Who's that person you're going to go talk to, and what are you going to talk to them about?
So in 1840, 2 German scientists, one of whom was Henle, proposed the germ theory of disease. Mm-hmm. Let's imagine that they didn't exist, okay? And I myself had gone back in time to 1840, when in the 19th century, the vast majority of human death and suffering was due to contagious disease. And I had written an article arguing for the germ theory of disease in 1840. Would that have changed history? Not at all. If Louis Pasteur hadn't existed, and I went back to the 1860s and 1870s when he was first becoming a biologist or a medical researcher, he was trained as a chemist. If he hadn't existed, could I have done better than he did? I don't think so. Now, in my own life, okay, mm-hmm. I've had the experience of going to aging meetings. I remember one in 1977, 42 years ago, when they were talking about, amyloid and the Hayflick limit for cell replication. I spoke to them as a voice from the future. I said to them, well, in aging, the fundamental cause of aging isn't one specific physiological failure. It's a whole bunch of them. Mm-hmm. Evolutionary theory shows us that. What you really want is to do research with an organism that lives vastly longer, that you can compare with organisms that don't. I said that all to them 42 years ago. Did that have any impact on them? None whatsoever. I published a paper in 1980 in this obscure journal called Nature, mm-hmm. outlining not only alternative evolutionary theories of aging, but specific experimental demonstrations of what was going on. That was the first time I published a paper in which I had longer lived organisms that I had created. Did that make any impact on biomedical research? Zero that I know about. So what have I done for almost 40 years since then? Just trying to get step 1 done. And to be very blunt with you, failing. Every single convert to SENS is another person lost to reality.
True.
It's like, I don't want to get too political here, but it's like the way I feel about Trump and his people. And I don't care whether you're a Democrat or what Bill Maher calls a classic Republican. Mm-hmm. I mean, he's the antithesis of the American Constitution. He just wants to be Mussolini, and his followers all want that. So every person in the American political system who's lost to the Trump movement is a person who is not going to uphold the values and integrity of the United States Constitution. Every person who buys into the latest cell molecular reductionist fairy tale about evolution is another person who's lost to reality.
I agree with you wholeheartedly. And that's why I think it's up to people like the show
Yes.
to
Yes.
keep these ideas alive, because and if I throw my point in there about this, I came from a pharmaceutical industry that has changed significantly over the last 35 years. There used to be hundreds of players, and now there's a handful that control that. Happening in the aging space, it's going to be disastrous. And unfortunately, I see that a little too often with the dynamics that are occurring. So that's why I want to keep the focus on folks like you that think differently and think with data and realistically, and not just, as you said, the whispers of God. So once again, I really appreciate you having spent the time to come on and talk about these topics. In the closing minutes, are there, obviously people can go to PubMed and read your papers, and go to Amazon and buy the books and read 55 Theses. Is there anything else that you want to close with? Get, other projects that we haven't covered? Please take the floor.
Well, one of the things that Aubrey de Grey often accuses me of is pessimism. And he does this because, from my standpoint, he's the Donald Trump of gerontology. And it's like you buy into his messianic message and all will be well. I think the evolutionary theory shows us something absolutely spectacular, which is also in the data if you look for it, which is that aging comes to a stop. Aging is only transitional between a period of very high health and a period of low but stable health. Humans on a modern diet get to that post-aging phase only rarely. So the last thing I would want to say is, contrary to the commonplace view, as argued by Aubrey de Grey, among others, the evolutionary view is not an argument for pessimism. It's actually the most radically optimistic view of what's possible in intervening with aging, because evolutionary theory and experiments with real organisms in my lab and others show that aging stops. Aging is just the transitory effect of the decline in the force of natural selection in the early to middle part of the lifespan. If you can survive that, you get to a plateau in which things stop getting worse.
Right.
Spent about 15 years working on that plateau from 1996 to 2011, culminating in our book, Does Aging Stop? If we control aging well enough, even with ad hoc tools that we could develop over the next 20 years, we could in fact produce the indefinite lifespans, the really marked control of aging that Aubrey promises and others promise, based on what is, in my opinion, entirely erroneous science, just by applying the insights that evolutionary biology supplies. There is a spectacular outcome out there, such that I would say that individuals born in the second half of the 21st century could live for centuries in quite good health. So I have a completely different route to the SENS type of route to indefinite lifespans. It's harder, it involves vastly more biological information, and it's based on what organisms tell us when you do powerful enough biological analysis. Long after I'm dead, people will be living for hundreds of years because of evolutionary biological research on aging.
I think it's an important topic that we need to, both of us, we're anti-reductionists, and we believe that that these other views really need to get out there, because we're not going to break through.
For you, Ira, So remember the geocentric, heliocentric controversy,
Absolutely.
right? Copernicus? Well, in fact, the geocentric theory is right in a small fraction of cases, which is the moon and the satellites that we put in orbit around the Earth. Okay? Otherwise, it's wrong. So in the same sense, the reductionist model is right for sickle cell anemia, Tay-Sachs disease, cystic fibrosis.
Okay, I'll give them that.
Yes. So you have to give them their small fraction of cases where they're right, but more than 90% of the time they're wrong. And by not accepting that more than 90% of the time they're wrong, they are responsible for the deaths of hundreds of millions of people.
Let's fix that.
Let's fix that.
Let's fix
Absolutely.
that. So we need minds like yours running the R&D show. Thanks so much, Michael. This was a true pleasure being able to talk to you for this time.
Thank you, Ira. I had a great time.
Thanks.